Think about the sheer mechanical effort required to pull water from deep underground all the way upward to the canopy of a massive tree. This diagram shows a plant's water budget. Despite the tremendous energy spent drawing that water out of the soil, the plant retains and utilizes less than 2% of it. The other 98% simply escapes. All of that excess water is ejected directly out of the leaves and into the surrounding atmosphere as water vapor. This process, the loss of water through the aerial parts of a plant, is called transpiration, and importantly, it occurs strictly against the force of gravity. This massive outward flow provides the exhaust for a highly regulated biophysical engine, essential for the tree's internal function. To find where this engine operates, we need to scale down from the entire tree to the microscopic surface of a single leaf. Transpiration occurs in 3 distinct anatomical zones across the plant: the lenticels, the cuticle, and the stomata. The lenticels found on woody stems, along with the waxy cuticle layer covering the leaves, account for very little of this process. They are minor, inefficient pathways for water loss. The vast majority of a plant's transpiration occurs directly through the stomata. The stomata are dynamic, actively managed exits that govern the plant's entire hydrological system. A single stoma is an incredibly precise microscopic structure. It consists of a central pore surrounded by 2 semicircular guard cells. The shape of these guard cells is dictated by turgor pressure, the internal fluid pressure of the water contained within the cell walls. When water flows into the guard cells, that internal turgor pressure increases. The cells become turgid, swelling up and forcing the central pore open. Conversely, when water flows out, the turgor pressure drops. The guard cells become flaccid, causing the pore to collapse shut. The plant throttles millions of gallons of systemic water loss simply by manipulating this localized cellular pressure at the leaf surface. The exact biophysical mechanism that drives this loss operates in a specific 7-step sequence. It begins after the roots have absorbed water from the soil and pushed it up through the stem. Looking at this vertical leaf section, water reaches the internal veins. It then moves into the loosely packed spongy mesophyll cells. These cells release a thin film of water on their boundaries, which evaporates into vapor. Finally, the vapor follows this downward arrow, diffusing out entirely. This final exit is driven purely by diffusion. The vapor naturally moves from the area of higher concentration inside the leaf to the area of lower concentration in the outside atmosphere. There are no active mechanical pumps pushing this exhaust out. The entire anti-gravity mechanism operates completely on passive concentration gradients. This raises a structural question. Is the exhaust system uniformly distributed across the plant, or is it strategically placed? We observe this regulation using a direct empirical test, where dry cobalt chloride paper is clamped to both surfaces of a living leaf. The blue paper turns pink when exposed to moisture. The results are clear. The bottom paper turns pink rapidly, while the top remains unchanged. This proves a distinct anatomical asymmetry. Dorsiventral leaves have evolved to concentrate their stomata almost exclusively on their lower surface. Plants physically shield their primary exhaust ports from direct sunlight, a structural adaptation that throttles the speed of the engine and prevents catastrophic dehydration. That physical shielding is necessary because the engine's speed is highly vulnerable to external weather variables. Increased ambient temperature and bright, direct sunlight both accelerate the internal evaporation rate, forcing the plant to lose water much faster. Wind velocity acts as another accelerator. High winds sweep away the stagnant water vapor surrounding the leaf, artificially maintaining a steep concentration gradient that pulls more moisture out. Humidity, however, has the opposite effect. Highly humid air creates a saturated environment outside the leaf that slows diffusion to a halt. To resist these forces, xerophytic or desert plants develop specific internal countermeasures, such as exceptionally thick cuticles or stomata physically sunken into deep pits. Leaf anatomy is an engineered compromise. It is locked in a continuous struggle to optimize its gas exchange function against a hostile, constantly changing environment. Zooming out from the microscopic mechanics of the leaf, we can see how this localized diffusion dictates the complete internal plumbing of the entire plant. The escape of water vapor out of the leaf causes a permanent drop in cellular turgor pressure at the very top of the plant. Because water molecules naturally stick together, this localized drop in pressure creates a continuous, uninterrupted vacuum effect that extends down the entire length of the xylem vessels. Looking at this diagram, we can trace that movement. This phenomenon is known as the transpiration stream or the transpiration pull. It is the silent evaporation at the absolute top of the canopy that physically drags water and deep soil nutrients upward against gravity. This brings us back to the opening paradox. Why would nature tolerate a system that inherently wastes 98% of its primary resource? The answer comes down to a biological absolute. A plant must open its stomata to absorb carbon dioxide for photosynthesis. It is a strict, non-negotiable requirement. Because the valves must open for that vital gas exchange, massive water loss is an unavoidable thermodynamic consequence. But the plant weaponizes this loss. It uses the massive upward flow volume to distribute heavy mineral salts system-wide, and it utilizes the evaporation itself to cool the leaves when exposed to intense sunlight. Transpiration represents an essential evil, a mechanism that converts the unavoidable vulnerability of gas exchange into a vital transport system for water and nutrients.